Okay, so we're going to make a start. So thanks very much for coming to one of our first in-person departmental seminars for a while. Today we've Bernhard here, who's one of the new associate professors and also tutorial fellow at Recoil College. Bernhard is going to be talking to us today about some of the neural mechanisms involved in memory recall. Bernhard's really made a big impact in this field. He's actually originally a graduate of I think it's Vienna, is that right?
The first degree,
yeah. And then he got his PhD with Vila Davachi in New York University. He's been working in Cambridge University in the UK, Birmingham University. He's told me that I don't have time to give a full list of all of his honours that he's received. I'm looking at his CV, I realise that's actually true. Bernhard, please.
Thanks very much, Mick. Let me just try these. Okay. Thanks everybody for coming. It's really great honor and privilege to speak in front of you. And let's make use of the in-person format and interrupt the speaker widely as much as possible. Ask clarification questions, philosophical questions. I really enjoy this dialogue. So I'm going to use this mouse here to cater to the audience at home. Hopefully this works for you. So let me start by giving you an intuition of what it is that we study in our lab. So we study episodic memory. And a good example is the inconspicuous question of what did you have for breakfast this morning? Sounds trivial, but if you really try to solve this question, you have to kind of mentally travel back in time, overcome an interfering lunch experience maybe, until at some point you almost feel like you can serve your memory of the breakfast. And then once you're there, I can probe many different details of your memory. So you might be able to tell me whether you had a sesame bagel or a plain bagel or donut. You might be able to tell me where you had your bagel and when you had your bagel. And these elements, the what, where, and when, are but a few examples of the manifold associations that make these beautiful associative memories or episodic memories. And the reason why I'm so intrigued by this phenomenon is that when it works, I would say it guides who we are as humans. Without our memories of the past, we wouldn't know who we are as a self, and we wouldn't be able to project into future challenges. On the flip side, when memories don't work for us, it can be quite devastating, as exemplified in all sorts of neurodegenerative diseases like Alzheimer's. There's even this weird phenomenon where you remember too much, like in post-traumatic stress disorder, PTSD. So given the importance of memory, both on the plus and the minus, I thought it's really important to get to the core of how this works in the brain. What makes life as a memory researcher a little bit more challenging is that memory is not just kind of a one-shot monolithic phenomenon you can study, but it consists of multiple stages. It all starts with the encoding stage, which is when you experience your breakfast, for example, for the first time, and try to commit this to memory. Then there's a so-called post-encoding offline period, which consists of both wake, rest, and sleep. And this is where we believe many of the fragments of our experience are solidified and manifested and stored more permanently. I'll talk more about this at the end of my talk. And the final part is what we just emulated a moment ago, when someone gives you a cue and asks you to travel back in time and reinstantiate or relive your past experience. And this is the recall part. And this is what I'll be focusing on today for you. So the big question that has guided my work for the last almost 20 years now is, how does the brain enable this lovely phenomenon of memory recall? And once we open the hood and look into the brain, I'm going to spare you the details of patient HM, but take my word for it, the hippocampus is important for memory. And so why might the hippocampus, shown here in a sagittal MRI slice, like if you were to cut through the head here, this pink region right here, why might this region be so important for memory? If you look at the anatomy, it seems to be in a very privileged position to integrate information from all over the brain. So it receives input from high-level association cortices, including object pathways, the so-called what pathway, including the where pathway, And it receives this through its neighbors in the medial temporal lobe, the parietal cortex, the perihippocampal cortex, which both funnel into the entorhinal cortex and then into the hippocampus. So the hippocampus is in a wonderful position to receive and send back information throughout the whole brain. So it's kind of a hub in that sense. And this anatomical oddity has given computational modelers reason to believe that the hippocampus can do a process that's called pattern completion. What is pattern completion? Pattern completion is the phenomenon where during the initial learning experience, you see different elements of the experience. Again, harkening back to the bagel breakfast example, you might perceive kitchen environment and the bagel in dedicated pathways. And what pattern completion does is that during recall, being provided with the word of breakfast or being presented with a visual cue, that information goes into the hippocampus again. Then it does its magic, the pattern completion process, also referred to auto-associative processes. And that allows you to then reinstate the missing link in the brain. So in a way, the way I sometimes think of it is as if the hippocampus were to receive a zip file of an experience in a compressed, abstracted form. And then once you receive a reminder cue, that zip gets unzipped and the whole experience gets reinstated in the rest of the brain, leading to this phenomenological experience of I recall my breakfast experience. That's the theory behind it. There are a few key features that we need to take into account if we want to tackle this phenomenon. First, cued recall or memory recall in general should be flexible. It shouldn't matter whether I ask you what you had for breakfast, what you had for dinner, if I show you the kitchen first, if I show you the bagel first, it should go either way. The hippocampus should be versatile enough to take a scene and reinstate an object, or to take an object and reinstate a scene. That's just one of the main psychological characteristics of cued recall. The second part is pretty fast. So behavioral data show that almost within a second, the mnemonic fate is sealed of your retrieval effort. So it doesn't take very long to reinstantiate your breakfast experience. And while I'm admittedly in love with the hippocampus, I do accept that it's necessary but not sufficient to do the whole reinstatement recollection process. So there's been a lot of great imaging work done that consistently identify a so-called core recollection network, of which the hippocampus is part, but it's not the only part. There are other brain areas, including the left parietal cortex, lateral temporal cortex, and frontal and parietal midline regions. So altogether, this makes up this core recollection network. So in the remainder of my talk, I'm going to try to pinpoint these three characteristics in an effort to elucidate how cued recall works in the brain. What do we know so far? Well, there's bits and pieces that can give us some hope that this is not a totally doomed endeavor. From rodent models, we can record directly from inside the brain. But the problem is when it comes to a kind of high level cognition, if you want to explain to a rodent the difference between familiarity and recollection, between remembering and knowing the real phenomenology of breakfast, you run into issues there. So it's limited. Same with us humanoids. You can do fMRI research, which gives us a great picture of where in the brain something happens. But when it comes to the speed of processing, this method lags behind, of course. It doesn't give us the temporal resolution to really decipher when these things happen in the brain. We can use EEG, sensors placed on the scalp, which is very fast and pretty encompassing on the scalp, but it's hard to measure from inside the hippocampus with that method. Or we can do something called intracranial EEG, which capitalizes on a rare opportunity to work with epilepsy patients and insert electrodes inside the human brain, record directly from the hippocampus, but then we don't see the rest of the brain. So we can see the forest for the trees, if you will. So all these methods converging provide bits and pieces, but none of them alone can really tell us how memory recall works. So in my talk, I'm going to try to mix and match a little bit of all of these human methods. I'm going to show you some fMRI data followed by intracranial EEG and finally some scalp EEG in the hope to put together all the missing pieces to make sense of how memory recall works in the brain. Okay, so the first part was done by a brilliant poster of mine, myself, more than 10 years ago. And the question was in an fMRI study, how do the hippocampus and the adjacent MTL regions, the neighborhood of the hippocampus, support its flexible memory recall? And it capitalizes again on the neighborhood structure of the hippocampus. So I mentioned these two players earlier on, the parietal cortex and the hippocampal cortex. The parietal cortex is an extension of the what pathway of the ventral visual stream, and the perihippocampal cortex is an extension of the where pathway, the dorsal spatial pathway. And so the reason to believe that these two areas within the medial temporal, so all within a small circumscribed region, code for object and scene representations. We can try to take advantage of this stimulus-specific coding to tease apart who does what in the medial temporal. So we started with an encoding, the learning part, which was pretty straightforward. So this is a paired associated learning task where participants see pictures of objects and scenes over one slide on one given trial. So this might be an image of a bagel in the kitchen, it might be an image of a teddy bear on an island, and so on. These are randomly combined. Your task as a participant would be to imagine these two entities together in real life or nature and tell me whether this is a plausible or an implausible combination. The answer to which doesn't matter so much to us. All I want you to is to imagine and bind these things in your mental imagery. Then we take people into the scanner and apply a cued recall task, where half of the stimuli, the cues, are the objects, and the other half are the scenes. And the task is very simple. You see a bagel, can you tell me what you saw the bagel with? You see the teddy bear, can you tell me what you saw the teddy bear with? And for the most part, we take participants' word for it, we trust them, but we don't entirely trust them, of course. We put in some catch trials where on some cases when they say, yes, I do recall, we have to verbalize what it is in the MRI machine, transcribe, and it turns out more than 90% of the times they are actually not cheating, which is good news. So there are different memory outcomes, of course. So you might be able to recollect what you saw the bagel with. So you might successfully recall the kitchen image, or you might not recall the paired associate. In other words, this yields a pretty straightforward 2x2 paradigm or design, where this is the only really awkward terminology in talk, I hope. So here in this terminology, I use the first letter is the cue, the second letter is the target, and in parentheses is the memory outcome. So OSR is this trial where you cue with an object, target is therefore a scene, and you recall the scene. An SOF trial would be your cue with a scene, target is therefore an object, but you're drawing a blank, you forgot the associated pair. So in this 2x2 design, we have 4 cells. So the cue type can be object or scene, and the recall could be successful or unsuccessful, recalled or forgotten. And these are the examples for each of these 4 conditions. And so we can use these 4 conditions to probe who does what in the medial temporal lobe. And we have a few reasons to believe there is a certain division of labor. I'm going to try to take you through what we think this division of labor might be. So if the parietal cortex is really the extension of the ventral visual pathway, it should care a lot about objects. So it should be on the scene, it should be active whenever an object is present, be it on the screen as a cue or in mind as a successful recalled target. So whenever there's these 3 cells, so these 2 because the object is a cue, and this cell because the object is successfully recalled, the parietal cortex should light up, if you will. Should show minimal activity in this case in the SOF trials because there's no object present, neither on the screen nor in your mind. You're drawing an object blank.
Sorry.
Yeah.
Just make sure I understood the task correctly. So it's all based on the verbal recall that they remember or not.
Yes.
So what's the advantage of this over having, I don't know, 4 alternatives, 2 alternatives, and then 3, 4?
It's a great question. There's a give and take in both ways. So if you, what I like about this scenario is that you can do trial unique images. So in what you outlined was a great alternative, have 4 alternative choices. You repeat the same objects or the same targets over and over again. Right? You can't afford to have a trial unique image on each trial. And so then you run into other related issues that you have to overcome interference. There's some kind of halo effect from one trial to another, maybe. So this allows us to be truly episodic in the sense that each experience is unique. At the downside of not having this verification every trial. If you express this prediction in fMRI activity or any other brain activity, if you plot activation in this cartoon fashion on the y-axis and the 4 conditions on the x-axis, this condition right here should yield the minimal amount of activity. Different prediction for parahippocampal cortex. To the extent that this area is involved in scene processing or representing scene information, it should be active whenever there's a scene on the screen, like in these 2 trials, or when you reinstate a scene by virtue of memory recall. But there should be minimal activity when there's an object on the screen as a cue, and you draw a scene blank. Finally, if the hippocampus is really just kind of auto-associated, as I sold it to you earlier on, we would expect the hippocampus to not care if it's flexible about cue-target relationships, but always be involved when we successfully recall information. So in these 2 trial types, because you successfully recall information, it should be active and should be minimally active when you forgot something. So very different predictions for 3 very adjacent areas. And this, 10 years ago, to be honest with you, this was not trivial because there were people around who would say, well, MTL is memory. The more you lesion in the MTL, the larger your memory deficit will be, but there's no clear division of labor. And here we came along having these very, very clear prediction of how subtly different these activation patterns might be. So these are this is the prediction again, minimal activity. So let me try to do this one more time. Minimal activity here, and these are the actual data, not as cartoony as I would like, but the only difference is the drop in activity when you don't have an object either or on the screen nor in your mind by virtue of memory recall. For parahippocampal cortex, the scene area, so this pattern, so activity whenever a scene is on the screen and when you successfully recall the scene, and minimal activity when there's no scene on the screen and not in the memory. And in the hippocampus, this was our prediction and these are the data. So I was quite happy seeing the pattern turn out the way we predicted it. And again, if you put these patterns side by side, it's not trivial. These are very closely related to each other, these areas. And if you plot their activity in this simple task, you can see there's a clear division of labor. Everybody in the MTL does something slightly different, and together they enable something as wonderful as reinstating the breakfast experience. So to summarize the first part, the hippocampus supports memory recall in a flexible fashion. We call it domain. General domain is just the same as stimulus or category, general fashion, drawing on domain-specific representations in the adjacent MTL cortex. So this is the part of flexibility. Yeah.
So if I understand it, it might not really matter whether I recall the correct scene or the correct object. If I said yes,
Yeah.
I would be able to tell the difference between me getting it right or wrong. But when I say no, I'm not recalling any.
Mm-hmm. Yeah, that's an interesting point. So in this scenario, we train participants, we instruct them very carefully to only indicate yes when they have a crisp mental image. They could pick the target image from an array of lures. So they really have a strong memory. And that said, they know that the target, so if you're cued with a scene, they know that the target would be an object. So they probably run through an internal database of template objects. So there will be some kind of attempt at object imagery. So it's not an entire blank you draw when you say I forgot, but the resolution of the image is not as crisp when you say I forgot than when you say remember.
Well, one funny bit on labels, and I remember what Teddy Bear was on.
Yeah, that goes back to the question earlier. I think there are 200 of these pairs shown to you, so that doesn't really get you that far. I think it would be an extra effort to encode the verbal label. The instructions are to only say remember when you're able to describe the image in great detail. And on those catch trials, they're quite elaborate. They say, you know, there's a kitchen with a nice picture in the background. So, but it's not all or none for sure. There's a gradient, and I think it would be interesting to quantify exactly how much they remember and see whether there's an area that tracks that scaling of fidelity of your memory. Okay, let's turn to the second criterion, the speed of mental processing for recall. So here we run, as I alluded to earlier, into limitations of fMRI. So we just don't have the temporal resolution to resolve what happens at the millisecond level there. So we did a different approach. This was in collaboration with Matthias Trader, a postdoc of mine back then. We turned to intracranial EEG. This is a so-called implantation scheme. So you can see a coronal depiction of the brain with electrodes coming in from the side to sample a particular part of the brain. And just to give you a little bit of background information on this procedure. So this is a clinical procedure. So we're working with epilepsy patients. Epilepsy is a neurological disease that's usually well controlled by anticonvulsive, anti-epileptic drugs. There are some cases that are so severe that the last resort is to go into the brain and remove, surgically resect the part of the brain that causes epilepsy. It's oftentimes just one particular focus, as it's called, that causes the seizures. And that is the biggest promise for these patients to alleviate the symptoms and be completely seizure-free. During that time, we as experimental psychologists can come into the hospital with our little experiments and offer to spend some time with us. It's usually a win-win for us. It's an amazing opportunity to record from inside the human brain. And for the patients, it's fun because they get to interact with scientists and they're just bored out of their mind, to be honest. They're sitting there for 2 weeks on the bed. I'll show you an image in a moment. This is done in collaboration with the Queen Elizabeth Hospital in Birmingham. And this would be a typical patient setup. So this is one of our previous patients sitting in bed, and what you might appreciate here is there are literally wires coming out of their brain going into an amplifier on the side that you don't see here. And here's a little side table that we hijack typically to put our laptops on and run our experiments. This is the setup. And this is the so-called post-implantation MRI image, shown here again on a sagittal slide and here on a horizontal slide. You can see what this implantation scheme translates to in real life. So you can see these holes sampled from along the hippocampus. These holes look a little bit more dramatic than they are because they contain a little bit of metal, which causes an MRI artifact. But making mistakes is definitely a very invasive procedure, and no one would volunteer to have this done just for the sake of science. We, for the study, recruited 11 participants, and this is kind of an overview of the coverage we had across all these 11 participants. So you can see a nice longitudinal coverage of the hippocampus right there. We applied the same experiment, the object scene memory task. Internally, we call it the obscene task because it produces obscene, robust findings. And it's entertaining for the patients too. With a little twist, at the end of the experiment, we added a so-called localizer task, where participants only saw pictures of objects and scenes and simply had to tell us, is this an object or a scene? It will become obvious in a moment why we put this in. But for the most part, let's focus on the encoding recall part. I'm going to show you data from this part right here, similar to the MRI study just before. So now I'm in a nice position to look at the timing of things. So the way I'm going to show you the data now is in the so-called time frequency representation, which displays on the x-axis time in seconds. Time 0 is when the cue comes on screen. On the y-axis, I'm showing you frequency, so we can resolve oscillatory phenomena across different frequency scales. This is from 1 to 150 hertz. And color-coded, in a second, you see the effect of successful versus unsuccessful recall. And it's open where anything would pop out, but what we found is this pattern. So just to orient you, again, color-coded is the kind of the effect, the recall effect. Warm colors means an increase for successful recall, and cold colors means a decrease for successful recall or an increase for unsuccessful recall. We're gonna get to the bottom of this in a second. One thing that's important to see is that within the first second, something seems to happen right here. So at about 500 milliseconds, in this higher frequency range, often referred to as the gamma range, there seems to be much going on when you successfully recall something as opposed to when you don't successfully recall something. It's quite striking, it's quite a clean effect. It's not the first time we've seen this. A few years before that, we used a different associative memory paradigm, also in patients. We saw pretty much the same effect right there. So let's look at this a little bit closer. if I plot the time courses for you, for the gamma, so this is now time average across these 80 to 90 hertz, just right here, in the heart of this gamma cloud, if you will, and plot the time course. You see again, time 0 is the vertical line. And see, no matter whether you successfully or unsuccessfully recall information, the hippocampus responds to the stimulus at about 200 milliseconds or so. So this, we probably can assume this is the moment when information goes from the eyes to the back of the head and again forward into the hippocampus and stirs up neurons in the hippocampus. But then the mnemonic fate is decided upon at about 500 milliseconds. So when you don't recall the paired associate, your activity in the hippocampus goes back down again. But if you do recall the paired associate, and this doesn't matter now if it's an object or a scene, the hippocampus stays on a little bit more. It doesn't just stay on a little bit more, but exceeds the activity from when you don't recall successfully. Between 500 and 1500 milliseconds or so. So this is a time where it seems very critical for memory reinstatement to happen. It's also just for completeness, because we're going to look at this a little bit later again. Look at the alpha time series. That looks quite different. So we see an initial bump for both memory conditions. Green is successful, gray is unsuccessful memory. Probably figured this out by now. And at about 800 milliseconds, hippocampal alpha power shows a further decrease when you successfully recall information. And in a moment, we're going to speculate more about what these two different frequency bands might signify. But let's focus first on the gamma cloud. The gamma frequency range has been linked to neural activity. So oftentimes in non-human primate work, spiking activity from individual neurons translates on a macroscopic level into gamma frequency increases. So this suggests that we're actually seeing individual neurons inside the hippocampus get excited and fire in response to the memory cue. And the more they fire, the more likely they are to reactivate this whole memory experience. So we had one patient where we could follow this lead a little bit closer, asking whether this gamma power is a viable proxy for individual action potential firing. And the way we did this was with so-called microwire imputations. So this was another epilepsy patient who volunteered for a special procedure where, in addition to the regular electrodes depicted right here, they volunteered to have a special electrode that would, at the tip, have a bundle of wires protrude. And this bundle of wires is useful in picking up single action potentials from the surrounding tissue. It's very rare to do this, and we only had one case study to do this. But nevertheless, I think I want to share this with you because it's such a rare piece of data, and it can inform us to some extent as to how much this gamma cloud, what is the mechanism that it symbolized. And so we looked at the data in this patient, and I want to believe this initial blip is just a noise piece of noise in the data, but what we see here is, again, green is successful and gray is unsuccessful. On the y-axis, I'm plotting now firing rate of a single neuron from inside the hippocampus. It's one cell. And again, we see it about 500 milliseconds. It increases no matter what. But if you successfully recall information, that neuron just stays on a little bit more heftily and a little bit longer. We can link the gamma power and these firing patterns more directly across different trials. We can say what frequency range tracks the firing pattern of this neuron. And this is the data I'm showing you here. So on the x-axis, we see now different frequencies, again, ranging from 0 to 150, just like there. And on the y-axis, I'm showing the correlation across individual trials with that firing pattern of the neuron. In other words, what frequency patterns track the individual firing of a nerve cell. And we see it's after stringent correction for multiple comparisons, it's exactly this gamma range between 55 and 110 hertz that survives that correction. So that gives us reason to assume that yes, this gamma cloud tracks single neuron firing from inside the hippocampus, sealing the mnemonic fate of cue recall. In other words, the hippocampal recall signal expressed in high gamma power or firing rates emerges at about 500 milliseconds. You can really set your clock to that. It's amazingly robust. Post-cue onset and distinguishes successful versus unsuccessful recall. Now, what's the functional significance? So cells being active, yeah, it's fine, it's exciting. But we talked earlier about this whole unzipping of information, of reinstatement of the breakfast experience. So can we link any of the hippocampus signal to that phenomenological explosion of recalling the whole breakfast? So the way I term this is, does the hippocampus serve as a switchboard that converts an external stimulus, the cue, into an internal mnemonic experience? So can we put some meat on this skeleton of theory of pattern completion, in other words? And so ideally, in a perfect world, I would have an electrode inside the hippocampus, one in parietal cortex and one in perihippocampal cortex, and do the same experiment again and see how information flows across these three areas. But because these are clinical patients, we can't choose where the electrodes go, so we have to deal with where electrodes are, and that's where this localizer from earlier comes in. So rather than looking at parietal cortex and perihippocampal cortex, we take advantage of the whole brain coverage we have in addition to the hippocampal cortex. This is one example. So the implantation is not just confined to the hippocampus, but spans many candidate areas in the brain. There's one case. Here's the coverage aggregated across 11 participants. So we get pretty decent whole brain coverage, not systematically so. That's a problem. That's why we can't easily generalize from one patient to another. But everyone has their electrodes in different cortical areas. And so now we take advantage of this localizer we include in this paradigm. We train a machine learning algorithm, linear discriminant analysis, to distinguish brain patterns that emerge when you experience an object from brain patterns that emerge when you experience a scene. So we basically train a classifier to distinguish the brain on objects and the brain on scenes. And this works quite well, very high classification performance. These are very potent, strong stimulus. So that's not too surprising. But then we would like to see what this classifier tells us, what kind of representation is present during the recall process. We'll expect maybe early on, when you're just concerned with the cue information, that the cue takes all of the space up in your mind. Then when you do successfully recall the target, then the target should occupy more of the representational space in your mind. So in other words, if this is again a cartoon, but if I were to plot classifier evidence for objects and scenes on the y-axis, pointing up as object representations and pointing down as scene representations, there are different predictions we can make depending on what kind of trial you're in. So if you're in an object cue scene target trial, you should first see the brain being immersed in the object experience, the cue. So first, classifier evidence should point up. Then if you successfully recall the scene, the internal representation of the memory should kind of push away the external information. You're not really interested in the external stimulus any longer. You're trying to elaborate, to answer Kate's question, you're trying to conjure up a vivid mental image of the associated scene. So the evidence should switch in favor of the cue to the target. Correspondingly, the same pattern for the scene cue object trials. First, scene evidence, and then followed by object evidence. And this first part, the cue-driven mental representation, should hold no matter whether you draw a blank or whether you successfully recall the target information, because you're just observing what's on the screen. But the second part should be especially prevalent when you do recall the paired associates. So it should be less prevalent in the forgotten trials. And so this moment where the lines of evidence change in favor of cue to favoring target presentation is a magical moment in the brain, in neural processing, because it's a moment when the internal representations override external information on the screen, to shift from the external world to an internal mind focus. And the question is whether the hippocampus has anything to do with this, of course. So here's the real data. Again, on the x-axis, I'm showing you time with respect to cue onset. Pointing up is object evidence, pointing down is scene evidence. For forgotten trials, we indeed see that for object cue trials, the blue ones, first, the brain looks like it's on objects, makes sense. And for scene cue trials, it looks like it's on scenes, which it should. but then nothing else is happening there. For memory transfer, we see this fantastic switch over. So at about 500 milliseconds, if you focus on the blue line first, you've just seen an object, but you want to recall a scene, and you succeed in recalling a scene. So kind of push away your mind's representation of objects and dedicate all your resources to scene information, and that's reflected in classify evidence. And the inverse for the other trial type without boring you with the details. So this moment, this crossover, is when the brain flips from a cue to a target representation. And the astute listener has probably taken note that this is exactly the time point when we saw the increase in hippocampal activity for successful recall compared to unsuccessful recall. I could even maybe convince you more if I plot or superimpose the effect size of the hippocampus in the gamma range onto this flip. This is exactly the moment when the hippocampus starts increasing, distinguishing successful and unsuccessful recall, is when the mind switches from external to a memory-driven representation.
I'm sorry.
Yeah.
On the two representations, so it seems that the two representations are not congruent. Have the evidence for one goes up, the evidence for the other goes down. Is that correct?
It is correct, yeah. But if in the case of the unsuccessful memory, you can see what happens. There's neither evidence for one nor the other. And so if one comes on, the other goes down, but the one that comes on has to come on very strongly to push down the other.
So when the representation, the codability of the target goes, let's say the blue line, the right side.
Mm-hmm.
So the codability of the one goes up, the Q goes up, right? And then what happens to that when the codability of the target goes down? So does it mean that it's, or what's the what's the midline?
The midline is undecided classifier. So you're plotting decision values basically, and the farther away from the decision boundary the classifier is, the more certain it is in favor of one or the other category. So if it's undecided, if the brain looks neither here nor there, it will settle on this chance line. But if there's strong evidence in terms of brain pattern for one category over the other, it will cross that line of indifference.
Does it tell you that it can successfully still decode the cue when there's more evidence for the target or not?
For that, I think you would need an independent third classifier. So you're right, this one is either or. But we can say that because it's below the chance line, the internal representation is pushed away, is stronger than the external representation. The external representation is probably still there, but it's trumped by the internal representation. If we had a third and a neutral category, we could see the two unfold in parallel probably. And probably seen the external representation would go down a little bit and the internal would go up a little bit. You're welcome. Yeah.
So I've got an online question here from somebody asking about that hippocampal peak and whether or not it happens slightly later. I presume they mean than that cross. Yeah. So what? Yeah. So the peak depends on which one you look at. So on the right, I'm plotting the effect size. So this is the difference between successful and unsuccessful recall in the hippocampus, and that's very true. It doesn't peak until about 1 second, but it starts changing, starts ramping up at about 500 milliseconds. So the way I think of this is that the hippocampus kind of ignites this pattern completion process. It's not just a single moment, but it's a process of kind of drawing evidence from all over the brain into the mind, and thereby just increasingly allowing the participant to conjure a vivid mental image. Okay, to summarize the second part, this is courtesy of Bettina, who did a great job as hippocampus the switchboard between perception and memory. So a hippocampal recall signal, high gamma or fine rates, emerges at pretty much exactly 500 milliseconds post-cue onset and distinguishes successful versus unsuccessful recall.
And this marks the moment at which cortex switched from perceptual cue representations to mnemonic target representations. He has a question in the back.
Hi, sorry. I was just wondering whether you've given any thought to that, the same spectral tilt is seen in uncued recall in the hippocampus, the same increase in gamma and the decrease in the low frequencies. So how does that then relate to its switching from perception to internal representation? Because the signature is very similar for uncued.
Yeah, it is, and I think that harkens back a little bit to Kate's question earlier. I think I'm selling this a little bit as all or none. You're either drawing a blank or you have full-blown memory recall experience. In reality, it's not. It's a little bit more continuous. And so you're in this artificial situation. You know there's a cue on the screen. It's going to be a target somewhere, and you have to find it. So you're going to deploy all your resources that you're aware of to try to retrieve the target information. Just sometimes you're a little bit more successful than other times. And this little bit more successful is reflected as a significant increase in hippocampal gamma or decrease in alpha power. And that little increase seems to mark the moment where the floodgates open and you have this recollective moment. But I think, what would be really interesting is to tamper with the system a little bit to see, is it really causal? What if you perturb the hippocampal signal at about 500 milliseconds versus at 200 or 800 milliseconds? Does this keep the phenomenological experience from developing? So there's more work to be done. But to go back to your question, I think it's once you exceed a certain threshold, then you attain this recall experience. Okay, last part. Once we said hippocampus is great, I hope you agree with me on that. But it's not all there is in the brain, unfortunately. So what are the cortical dynamics during successful recall? So how does that, I've been talking about this mnemonic fate and phenomenology of recall unfolding, how does that look in the brain? And how does this whole core recollection network that's always shown in fMRI studies, how does that come into play? So now we switch to a third method, to Scalp EEG, high density with 128 channels and 20 participants. And same idea, I'm not going to bore you with the experimental details, but we again contrasted what happens in the brain in Scalp EEG for successful versus unsuccessful memory recall, same remembering versus same forgotten. And here's what happens. I'm going to take this apart a little bit more, but just to give you a first overview. This topography we can easily ignore. It's going to get more interesting when we're looking inside the brain, but just for completeness. But here it's getting a little bit more interesting. So this is again a time frequency plot, now only ranging from 1 to 50 hertz, because at the scalp you can't really resolve those higher frequencies that we're also interested in, gamma frequencies. And you can see if we kind of collapse across time, you can see that the most interesting frequency band is again this alpha frequency range between 8 to 12 hertz. And if we collapse across frequencies, time window of interest is just starting at 500 milliseconds, maybe peaking at 1,000 milliseconds or so. Kind of a first approach to the data. But then let's look under the hood, do some source localization, which is always to be enjoyed with a pinch of salt because it's very, it's analytically complex, it's everything but straightforward. But you can kind of mathematically try to derive where the signals that you observe on the scalp might come from inside the brain. And so this is what we did. We collapsed across this time window of interest between 500 and 1600 milliseconds and saw where does the alpha power show up in the hope that this roughly resembles the fMRI results. And it did, to a positive surprise. So this is now shown on a kind of default inflated brain. It's not a perfect match, of course, but you can see nicely that with EEG alpha source localization, you can replicate this left angular gyrus, parietal cortex part, see some midline evidence, just like here, maybe a little bit of hint of prefrontal cortex, and something that looks a little bit, if you're so inclined, like the hippocampus, which is of course outrageous to claim because with Scalp EEG, how can you possibly localize the hippocampus? But then with all these convergence, so if you now look at the time course, first of all, I should say this is also not the first time we've seen this. So from a previous study, we used MEG, so slightly different experiment and different results. But again, we can source localize alpha power to these cortical core recollection networks, which almost might put fMRI out of business, at least for the memory client researchers. Anyway, we can look at the time course of how this core recollection network comes on the scene, when which player enters the scene, so to speak. And the first entry we see at 800 milliseconds, again, in the MTL. And again, you can't really say too much about hippocampus because it's all from the source. But mind you, this is exactly the time point where in the intracranial study, where there's no ambiguity about the source, this was from inside the hippocampus. So the same frequency range, 8 to 12 hertz, the same time point, where successful and unsuccessful memory diverge. So again, I'm not suggesting we sell all our MRI machines because there's lots more work to be done there, but it seems to suggest that at least if the psychological effect is pronounced enough, and if the signal, the alpha power, plays ball with the researcher, we can source localize some of these intricate memory processes inside the MTL. Yes, Irene.
Sorry, good question. You know, at the beginning, the alpha sort of goes up.
Yeah.
And also in the intracranial.
Mm-hmm.
Do you know why that would be? Or like, do you have any guesses?
No, I don't know. What I think is that my speculation is that the ERP leads into that frequency range. So whenever you show something on the screen, the local field potential just goes crazy, and the first couple of milliseconds studies pronounce components with sharp transients, and they bleed definitely into the theta, but also in the alpha band. So I think that's it. Okay, but now we can observe, we can kind of play a movie, how this core recollection network unfolds in the context of memory recall. So we see what the fMRI data wouldn't show us, that after the MTL, the posterior parietal cortex comes into play, followed by medial prefrontal cortex. And there are members in the audience who have way better theories about what the medial prefrontal cortex might do. But one very hand-wavy idea is that the medial temporal lobe provides a recall signal, maybe the first line of evidence for some memory traces. Parietal cortex serves as a buffer function, kind of almost like a working memory storage to manage all these pieces of evidence. And then prefrontal cortex deploys the action plan, remembering or forgetting, and integrating the whole posterior evidence into a motor response. But that's all for discussion. And then the next one. So successful recall encompasses a signal cascade from the MTL or the hippocampus even by the posterior parietal cortex to medial prefrontal cortex. And that brings me to the end of my talk. To conclude, I came before you to talk about the hippocampus and pattern completion. And I'll show you some evidence suggesting that the hippocampus supports memory recall in a flexible or domain general fashion. That the hippocampus recall signal expressed in high gamma power or individual neurons firing rates emerges at about 500 milliseconds after you've been presented with a cue like breakfast or anything else. This seems to mark the moment at which the brain switches from perceptual cues to mnemonic targets and triggers sequential engagement of posterior parietal and prefrontal recall networks in service of memory recall. Before I call it quits, I just want to give you a little preview of what happens next time I'm going to speak to you. The last couple of years, we've been focusing on these offline periods. I just want to share with you some recent accomplishments. When we turn off the light at over turns into overnight, and we just finished implementing a sleep lab there. So this is my lovely lab, Marcel, Lena, and Jude. And this could be you. Look at how happy Jude is. Sleeps like a baby. We see nice slow waves and spindles, but that's for next time. So with that, I just want to thank my lab again and thank you for your attention. Are you going to do the MC?
I'm happy to do that, but if you want to do it yourself.
Oh, good.
Fate, I think you'll present first.
Super thought, thank you. I was actually wondering about, it may be a weird question, but your encoding trials, you had this plausible versus not plausible. Did you ever look at the, I mean, was the data we were looking at just plausible scenes, or was it everything? And
It was everything.
Did you ever look at the split there?
Yeah, it doesn't, so it started very kind of just looking at some tasks that would engage participants, but it turns out it actually makes a big difference. So if you say it's plausible, then you're much more likely to recall information. So the good thing is it spreads evenly between object cues and scene cues, so there's no confound there. But behaviorally, it's definitely the case that whatever is deemed as semantically congruent or plausible has an easier way into memory. And there's a few interesting follow-up things I would like to study, especially when it comes to consolidation, maybe things that are more schema consistent, bypass the hippocampal encoding necessity and so on. So there's more work to be done there. I would have liked for it to show no effect at all, just have a kind of incidental encoding condition, but there's more behind the scenes there.
Yeah, because I mean, I guess that means then your split on forgotten and remembered are a little bit also kind of what was presented plausibly and not plausibly as well. That's
true. Yeah, that is very true. But I have looked just for my own peace of mind at just the plausible that were remembered and forgotten, and you see the same pattern. So there's no interaction there.
Okay. Sorry if I'm taking too much question time, but just as a follow-up, because I guess like my root of this question was also in this idea that it's really switching. What you're doing is really switching between perception and memory rather than kind of integrating between perception and memory. And I wonder if in these kind of remembered cases, what one thing you can also be doing is taking in just like a lifetime of experience with bagels and where bagels should be compared to, I don't know, some weirdo object that has no right being in the kitchen. And how much that might kind of feed into that effect, whether it's really this strong kind of episodic this moment, or whether it's this kind of, I don't know, combination of many.
It's a great path you're taking.
Sorry.
No, I think it's, so this is of course very simplified, this whole like 1 second cue recall. I would predict in a more realistic scenario, much more akin to the work that you're doing, for instance, there's a more dynamic process. First, talking back and forth between perception and memory. The more complex the cues, the more you need to look back. I would predict that certain oscillations might help toggle back and forth. So it gets much more interesting when you have more realistic and naturalistic stimuli. But this is just, the most simple version, and maybe that's why we see this nice. You don't have to remember the cue anymore once you reinstate the target in this case.
Yeah.
But in case where you do, I think it's getting more interesting.
Cool. Thank you.
I wasn't clear whether you think the hippocampus is in a representational function or some sort of decision making function. So if you had an auditory object like the sound of a seagull against a scene of the seaside, would you get the same effects, or so are the representations modal, amodal? What's your thinking?
It's a wonderful question. In the first instance, I would say they're amodal. From what we've seen so far, it doesn't really matter what kind of stimuli you use. Here I've shown the flexibility between objects and scenes, but if we were to use sounds or smells, it shouldn't really matter because the hippocampus serves as this hub for kind of multimodal representations. But unfortunately, it's not as simple as I would like it to be. When you look inside the hippocampus and try to capture individual neurons and see what they code for, there's an interesting salt and pepper mix in the hippocampus. Some cells seem to be agnostic as to what the stimulus is. They're just concerned with the process of pattern completion. But there are also other cells that very much care for what stimulus you're using. And they seem to be more tuned to scenes compared to anything else, which relates back a little bit to the whole rodent work on place cells and spatial navigation being an important function in the hippocampus. So it's unfortunately not as clear cut, but there are cells of which I'm confident that they don't care about what it is that you associate. Those are amodal, and they just serve as the indices to where information lies in that unzipping of information.
I just want to pick up on the duality that you were trying to make between perception and remembering. And so I think there are a number of studies which indicated that as you pair two things together, regardless of it's an uncanny combination, the kitchen or whatever, the patterns of activity become similar with each other. So it seems that the brain, which means that for your decoding, it becomes even more difficult to decode A from B, which are paired together. So it seems that this similarity patterns is kind of in odd with this duality of perception against remembering. How would you explain that?
Yeah, it's another great point, and you're putting the finger where it hurts, unfortunately, because yeah, I think we're simplifying this external cue, internal target, whereas in reality it is much more interwoven, both dynamically, going back to Sage's question, but also in terms of representation. So in real life, if you learn a new concept, different elements of that concept merge together, and it's not so clear cut what is cue, what is target, what is cause and consequence. And there have been actually some good single unit studies too, by Rodrigo Quiroga's group in Leicester, showing that as you have multiple repetitions of the same experience, one cell kind of stands in for another representation. So you can almost rewire certain cells. Because this was just a one-shot learning, a single exposure, I think we run the risk a little bit less of that. But I think the experiment to do would be to see if you have multiple exposures and overtrain people in certain representations, whether probably using RSA and not decoding, whether these representations merge more and more. And again, what is consolidation to kind of changes in representation, but would be good to tackle at some point.
Great talk, by the way. So you talked about this replay mechanism during sleep, so this offline and remembering the past event. I'm just curious whether you can disambiguate successful and unsuccessful recall during sleep, like when you're trying to replay again.
Yeah, it's a fantastic question, Junior. Thank you. It's experimentally challenging because you don't have the luxury of just presenting a stimulus on the screen. So you kind of, what you can do as a sleep and memory researcher is you can attach electrodes on or inside the brain and observe what happens. And then the hope would be that experiences that are better remembered or better encoded show more evidence of coming back during the night. So for instance, we're running a study at the moment where you come in on 1 week and I just have you learn objects, and you come back another week and I just have you learn scenes. The idea would be that after having learned objects, replay would suggest, again, object information coming back. And after the scene learning session, scene information will come back. I have to keep you waiting for the results, but there's another method we use, which kind of takes back a little bit of experimental control, which is called targeted memory reactivation. So during learning, you link every experience to a unique sound, for example. And then during sleep, you play some of those sounds and leave other sounds unplayed. And what we then see is that the sounds that were played during the night, even though the participant doesn't remember or is not aware of that, those bits of information are better remembered next morning than the ones we don't reinforce. So I think there's reason to assume there's both endogenous reactivation of successfully learned information, and we can exogenously tamper with the mnemonic fate of experiences overnight.
One question I was wondering was, you're using objects in spatial scenes because that seems like a very natural model to have for an episodic memory. That's one, beginning you were talking about. That's one of the things that maybe defines an episode, part of what defines it. But one of the things that you didn't do was show experiments where you look at learning associations between pairs of objects or pairs of spatial scenes, and I can see that they don't give you some of the same analysis approaches, but do you think the same sort of things are happening there? Is the hippocampus important in that type of association, or
yeah, it's a great question, and it's something that's been troubling me for a while. So first, I think even objects and scenes are not as well behaved as I like them to be, because every scene is made of many objects, but not every object is made up of scenes. So there's an asymmetry there. And there was a little data point where I said the panoramic cortex is not behaving so well. I think it's exactly
right
because of that, because it also cares for scenes, because it latches onto the object in scenes. That was another question. There's a good theoretical model by Andrew Mays at Manchester who says there's a difference for the hippocampal, for the need of hippocampal involvement for within domain associations versus between domain associations or cross domain associations. Within domain would be pairs of objects, pairs of scenes, and cross domain would be object and scenes, faces, and so on. And the idea there is that the more distant the representations are in the brain, the more of the hippocampal pull you need to bring them, to unify them. I haven't tested this experimentally, and I think there's all sorts of kind of unwanted confounds in the sense that you have maybe more interference if you have object and object. The mental image is not as clear cut. So I think the jury's still out to really map what kind of pairs, abstract, complete, within domain, across domain, drive hippocampus. But my idea is that so long as representations are not unified, they're separately represented in the rest of the brain, you need the hippocampus as an index device to pull together that information.
okay? So then one last question, this one was posted online by Jeff Bird. So he was curious whether or not any of the later activation changes that you were seeing. So, for example, in the movie that you were talking about in the one of the final slides there, some of the areas in the medial frontal cortex, would you hypothesize that those were things that were important for metacognition and memory?
I think so, and again, that's where I would defer to more suited researchers to address this. But yeah, I would think so. I think metacognition, you're in this situation where you might have to report on your mnemonic outcome. You have to decide what action to deploy, which button to press in the MRI machine or in the EEG. So it's kind of integrating all this mnemonic bottom-up information into a novel action plan. That's my very hand-wavy textbook cognitive neuroscience explanation. And I think each of these hubs would need to be a little bit poked to really understand the exact contribution.
Okay. Thanks very much. Okay, thank you, Bernhard.
Thank you.